All right, so now we get into the topic of evolution. And when thinking about evolution, we're talking about basically how life originated on this planet. Now, many times we think of evolution as just the evolution of man, but we have to stop thinking of that, and we have to start thinking of evolution as just life in general. Like, for example, how did life on Earth begin? Obviously, we know we weren't the first signs of life. So there were, plant life, bacterial life, animal life that came way before man ever did. What were some of the earliest forms of life? And when these lives finally began to exist, how did they start to interact with our biosphere? Now, these are questions that even till today we have very strong theories that we believe, and it's a story that is changing, and we really don't have concrete answers to this. Now, we do have, in speaking of theories, we revert to cell theory throughout thinking of all of this. And the three components of cell theory are that cells are the basic unit of life, that all living things are made of cells, and then one of the more vital ones is where cells come from pre-existing cells. This is called biogenesis. Now, the one question that remains completely unanswered till this point, and again, like I said, we've got our theories, but nothing is concrete at this point. But the one question that comes about here is that if cells come from pre-existing cells, and just so that we understand what that means, is that cells come from another cell that existed before it. So cells come from another cell that existed before a pre-existing cell, then the question is going to remain, well, where did the first one come from? That brings to light that whole what came first, chicken or the egg conversation. Well, in thinking of cells, if cells come from other cells, there had to be a first cell that came from somewhere. How did that one come about? How did life begin on Earth? Now, like I said, we have a few hypotheses that we follow strongly today. We have a very small amount of evidence, if you kind of think about it. The evidence that we have on this is kind of small. It's not really a lot. And it leaves us with some gaps and uncertainties as far as how life originates, and not just originates, but also how life changes over time. But one of the biggest ideas that even till today that we follow regarding this whole entire topic is the experiment that comes about with Miller and Urey. Miller and Urey were two scientists that 50, 60 something years ago, back in the 50s, well, they started their work back in the 30s actually, and really got together and did some huge strides in their work in the 40s, and it wasn't until the 50s that they actually took a huge step forward with their work. This picture that you're seeing right here, which you're probably going to see on the EOC, is huge. It's extremely important, and it doesn't give us the answer that we were looking for, but based on their work, it set us in a particular direction. You see, what they wanted to try to do is not to actually prove how life began, but they wanted to try to see if they could recreate Earth's early atmosphere when life did begin some 4 billion years ago, which is about 2.5 billion or so years after the planet began, again, in theory, what we believe. And it took a few millions of years for the Earth to finally cool off. And so when that did, that kind of stabilized, again, over the course of millions of years, it kind of stabilized Earth's atmosphere. And when it did so, this is what they wanted to try to recreate. So they did this experiment, and they saw that Earth's atmosphere, we strongly believe, to be almost absent of oxygen. There's little or no oxygen in Earth's early atmosphere. It was mostly carbon dioxide, water vapor, nitrogen. We had some lesser amounts of carbon monoxide, hydrogen sulfide, and hydrogen cyanide. Obviously, today gases that are considered very toxic. So our early atmosphere compared to today was a very unbreathable atmosphere. So they created this experiment, if you will, this sort of contraption to help recreate Earth's early atmosphere. Again, in light of trying to search for an answer of how life began. So let me explain how this experiment kind of works, the very fast version of this. First, they take water and they heat it up. Now, the reason for that is because you're trying to create water vapor. Water vapor was very, very big in Earth's early atmosphere because Earth was basically considered for a very long time a very large ball of active volcanoes, basically completely engulfing the entire Earth's surface. Now that puts out a lot of water vapor out into the atmosphere. So they took water, heated it up, create the water vapor. And then through this part, they mix in methane, ammonia, hydrogen, added to that water vapor, because these gases were believed to comprise a good portion of the Earth's atmosphere. Now the water vapor, now mixed in with these chemicals, now comes over to this part of the experiment where they put these electric components to it that would basically, they were like electrical nodes, if you will, that would release sparks of electricity into this circle. And what they were trying to recreate here was just basically the electricity produced in lightning and storms way back in Earth's atmosphere at that time. This is your chemical spark. So these electrodes, these nodes that are here, are what basically create the spark that basically pushes that chemical reaction to occur. Water then is passed through this condensation chamber. So basically water is cooled off again to basically form back into water droplets. These water droplets, the condensation forms here, eventually accumulating into droplets, and water would basically drop down into this container and settle. And after about a week, they found amino acids in that water. These amino acids are actually believed to be the building blocks of protein. And again, proteins are building blocks for life. So Miller and Urey devised this experiment here, which again, you will see throughout the year, maybe throughout the year in several tests and so forth, and we'll learn about it in class, but you'll most likely also see it on the EOC, very important as well. And they did this to try and see the Earth's early atmosphere, and what they found was amazing, because they saw after a week this liquid collected these amino acids, which can contribute to the building of proteins. Now, like I said, it took all this time for Earth's surface to finally stabilize. 4.5 billion years ago is when 4.2, so the Earth had already existed for a few million years. Earth finally cooled off, where the surface of the planet can basically harden, rocks would form. And this is important because this is what allows our ocean to form. When the Earth's surface was finally able to stabilize, our oceans were able to form and take place. Again, going back to their experiment, in 1953, I told you it was in the 50s, in 1953, Stanley Miller and Harold Urey tried to recreate Earth's early atmosphere, and they tried to answer a particular question which they actually did not, but in doing so, they produced, or their experiment produced, the 21 amino acids that were the building blocks for proteins. Now, Miller and Urey's experiment gave us an avenue, if you would, a different direction toward more questions. They tried to answer a question, and what they did is create more questions. Here's where we get into their work, is what leads us, or led other scientists to get into the origination of the cell. Remember, this is all about how life begins. And so if you get these amino acids, which can make proteins, and proteins together can make cells, now you're getting into that direction. So their evidence basically suggests that after Earth cooled off, enough of that liquid water that cooled off when it went through those series of motions of the water vapor from the volcanoes mixing together with the atmosphere, with the atmospheric gases, and then your storms would ignite these chemical reactions, and that liquid water would eventually cool off, and it would carry these cells that are similar to bacteria. They weren't exactly cells, but they behaved like cells. They were called microspheres. Okay, these microspheres were basically like cells because they developed, or not sorry, they developed. They demonstrated some of those eight characteristics of life that we've gone over. They demonstrated some characteristics of living things. Now we're talking about getting inorganic material and creating organic material from it. At first, when this liquid water carried these cells similar to bacteria, these large organic molecules formed these tiny little bubbles. These little bubbles are called protenoid microspheres. Those are the microspheres that we're talking about, that they're not exactly a cell, but they demonstrate some of the characteristics that cells do, of living cells. So protenoid microspheres were what we found in this liquid water to be very common back then. So now these protenoid microspheres carried what we call organic molecules. They were the formation of organic molecules. They carried things in them like the amino acids, sugars, fatty acids, and they carried nucleotide bases, okay? The origins for DNA and RNA, okay? So you're getting organic molecules from inorganic molecules. Remember, the protenoid microspheres were inorganic, but they produce amino acids, sugars, fatty acids, and DNA and RNA. Now you're talking about organic material. So getting organic material from inorganic material, that's known as abiogenesis, okay? This is what Miller and Urey gave us the answers to. Everything used in their experiment was inorganic material. They used water, they used gases, the electricity, and they got out of that the amino acids that could be combined to make proteins, that can be combined to make fatty acids, and DNA and RNA. Excuse me. So let's talk about a little bit about DNA and RNA. They're the same way that we want to know what comes first in terms of, how life finally came up. We need to also understand what came first with cells, whether they had cells with DNA or whether they had cells with RNA. And the RNA world hypothesizes about the origins of life, suggesting that it evolved before DNA did. Remember that RNA, to a very simplistic manner, can be thought of as just a one-sided copy of DNA. But again, in thinking that, there are several steps that could have led to DNA-directed protein, the formation of DNA-directed protein in this as well. So, we don't have the exact answer as to all of this. Remember that in science, pretty much everything is a theory, but we have our very strong beliefs, and so we do believe that RNA did come before and did evolve before DNA did. Remember that cells are controlled by the information stored in DNA, which is going to be transcribed and translated into RNA, and then translated into a protein eventually. Okay. So obviously that would imply that you're not going to have DNA before having RNA. So, going back to our first cells. Cells, remember, organic molecules have a tendency to aggregate, to aggregate together. Okay? And so when our first cells did this, we have to try to find out how and why. Cells developed a kind of layer around them. It's called a phospholipid bilayer. And that phospholipid bilayer is our first formation of a membrane, basically. It's where lipids, in their avoidance, in part of their avoidance of water, line up in a particular way where they form basically two rows of lipids. That's where we get the term lipid bilayer. And they form two rows where the head part of the lipid, which is okay with water, which likes water, faces to the inside and outside of the cell, but leaving the tail of the lipid to the interior inside and the interior outside of the cell. And this is the part that doesn't like water. We refer to that as being hydrophobic. The cells basically formed this first lipid bilayer because it was, remember, surrounded by water on the outside, and the cell has water on the inside. So that's why you need that head portion of the lipid to face outside, and then another head portion to face inside, so that the tails, which are hydrophobic, tend to not be exposed to direct contact with water. So as a result, this membrane starts to form, a membrane-like vesicle. We call those coacervates. And these coacervates form real easily under these certain conditions. These are kind of like thinking of that coacervate as, you could kind of think of it as a cell, but not a cell. It has some of the things that cells form, like those lipid bilayers, but they weren't exactly a cell. Coacervates are basically made of these aggregated, these compressed together lipids. They just simply expand by just simply through polymerization. And when they reach too big of a particular size, they would just simply split. So you take, here are these lipids. So I talked to you about the head of the lipid and the tail of the lipid, as you can see here, right here in this part. You have the head right there, and you have, here's the head. I'll draw over this one. You have the tail right there. And so what they do is they basically form all these various, and again, I'm sorry, I'm kind of slow on this, but they form all these heads with the tails facing this way, and then on the opposite direction, you get them facing the opposite side with, again, the tails facing in. And I went too high on my drawing, but you basically got water on this side, and you have water up above on that side, and you can't see it. So here we have lipids forming by means two layers together. Okay, that's your lipid bilayer. Now water passes through here. Water does pass through here, through this membrane type structure. Water just doesn't stay in the middle. So the tails are not exposed to that so much. The head of the lipid is. Now, because they compress together, they aggregate here, they form semi-permeable, a semi-permeable membrane, which means water can pass through, but not completely pass through. All right, so it's semi-permeable. It holds water out, but yet does stay enough to allow water through. When they aggregate together and they form these layers, it will form this structure, this coacervate. The coacervate will continue to go through polymerization, which is its growth, until it becomes too large, and then it will just simply, this side here will join to the middle of this, of this side here, and what you get is that this part comes in, and this is basically what you get. This will eventually unite here and split apart in opposite directions to form two. And then it will go through the whole entire process, again through polymerization, all over again, and will continue to do so. Now, the hypothesis basically suggests that these structures, similar to microspheres, might have acquired more of the characteristics of living cells. Remember, coacervates are not protenoid microspheres. They are, we're talking about the protenoid microspheres as having the structures for the amino acids, and then this is where the coacervate basically gives the idea of how they are going to start to copy, and the formation of the membrane as well. Now that's important because the formation of that membrane, which I'm going to address now in a couple of slides, develops what we call a nuclear envelope around the nucleus of the cell, the development of that nucleus, the eukaryotic cell. So now, in talking about those origins of life, remember, we're talking about cells or structures that look like cells, behave like cells, but are not exactly constituted as cells. Now, one of the characteristics of life is that all living things contain that hereditary information. So this is where that polymerization becomes important, because this is how you pass on this genetic material from one to the other. Now, we believe that RNA may have actually been that first hereditary information to pass through. And there are several hypotheses as to how RNA could have evolved into that cell. This is what gives us our first eukaryotic cell. In 1967, a scientist by the name of Lynn Margulis developed what we refer to as the endosymbiotic theory. The endosymbiotic theory is where she, to date, has basically done, probably considered by many to be the best job possible in answering that question. That question that we talked about if all cells come from pre-existing cells, where did the first cell come from? Well, this kind of touches bases on that. She proposed that the eukaryotic cell comes from the formation of various things, basically forming together through the prokaryotic cell. So according to her theory, eukaryotic cells form from a symbiosis of several different types of prokaryotic cells. Remember, cells without a nucleus. So basically, her theory suggests or kind of works like this. You have your aerobic bacteria. This aerobic bacteria is actually going to end up becoming an organelle in this cell. Now, here you have your anaerobic prokaryote here. You have a cell structure that has no nucleus. Remember, the no nucleus part is important because, remember, we're talking about passing on genetic information. Through passing on genetic information, that's how the cell is able to understand, and I almost hate to refer to it that way because they don't really understand, but you know what I mean. But this is how the cell basically gets to the point where it can copy, and that the copy has a set of instructions to basically allow it to take on a similar life pattern and continue to copy when it needs to copy. So, okay, going back to this, you have your anaerobic prokaryote, okay? Anaerobic meaning not requiring oxygen at this point. You have your aerobic bacteria. Here's the induction of oxygen into this thing. And like I said, this bacteria eventually becomes organelles inside of the cell. Specifically, since we're talking about a bacteria, it has energy. So the organelle that it basically becomes is the mitochondria. Now, in here, when this is going on, you have your nuclear envelope evolving. This nuclear envelope is going to eventually become the nuclear membrane, if you will. At the same time that all this is going on, now you have your aerobic eukaryote, because now you do have the formation of your genetic material with your nuclear envelope here to pass on RNA into this. And now you have your mitochondria. You're basically your second organelle that starts to form. Through this, you have your photosynthetic bacteria that starts to come in. And now and little by little, more and more organelles start to become of this. This eventually develops into cells that led us into the line of plants and plant-like protista or protists. You have your photosynthetic eukaryote at this point. Now at this point, from here, you have your plants and your plant-like protists. Without your photosynthetic bacteria, cells take off in a different direction. Your aerobic eukaryote, which now these will eventually become cells that form animals, fungi, and non-plant-like protists. So, at this point right here, the cell develops in two different directions. Those cells that acquire photosynthetic bacteria eventually become plant-like cells. And the cells that develop the use of oxygen, okay, or the necessity and use of oxygen, and start to thrive off of that, start to eventually become our animals, fungi, and our non-plant protists. And so basically what she had thought of was, you have your protenoid microsphere, you have the induction of other types of bacteria that become part of the cell. They're basically engulfed into the cell. And the cell evolves in two different directions here. Photosynthesis being the primary deciding factor between the two. And through this theory, you have plant type cells and animal type cells eventually. Lynn Margulis's endosymbiotic theory is basically, to put it in a very, very, very simple term, the combination of various types of bacteria with those little bubbles that we spoke about called protenoid microspheres, and that is the development of the first cell. And from there, cells just continue to evolve and evolve and evolve until we get what we have today.